name is Stephanie Watts. I'm from Michigan State University in the Department of Pharmacology, and I'm joined by Dr. Andres Contreras, who is from the Large Animal Clinical Sciences Department, also of MSU. We just got done doing a session, Recent Advances in Hypertension, that was focused around adipose biology. So I was the one to organize the session, and as I said in my introduction, I really organized this purposefully and deliberately. So the group of people at hypertension know a lot about high blood pressure regulation, and many of them have begun to touch more on how adipose tissue or adiposity changes the cardiovascular system. That said, though, they're not experts. Right? They don't know the ways to study adipocytes and that there are different types of adipocytes or different mouse lines that might be useful, or a big picture of how adipose biology or adipose tissues might change the cardiovascular system. So we had 3 speakers this morning that epitomized those ideas that I just described. Dr. Christy Gilniak, who is in Philip Schur's group, speaking very much about the adipocyte, that there are different types of adipocytes, and how hypertension and obesity might change how those adipocytes work. She described a triad which include inflammation and fibrosis and metabolism as something that means that the quality of the fat you have is more important than quantity. So she educated us in regard to that. She was followed by Annabelle Wong at the City of Hope, who has some fantastic tools to be able to understand where fat cells come from. These things are called adipocyte progenitor cells. And she demonstrated convincingly that there are some cells that we may not have known existed, that as a body ages, they grow or they expand or they are utilized in a way that makes an older person gain fat at the same time that they're losing muscle mass. So to a group of hypertensionologists, that's important to be able to know. Right? The last speaker, and I'll let him talk for himself, is Dr. Andres Contreras. So Andres, do you want to tell us about what you did and then how that linked with the other 2 speakers?
Yeah, thank you. So I summarized the concept of adipose tissue in the context of cardiovascular disease and cardiovascular function. 3 key things in my talk. First, that adipose tissue is everywhere and that its anatomical location matters. So there are specific adipose tissue depots that might influence or might increase the risk for cardiovascular disease because just their location. And I gave some examples, the fat in the kidney and the fat around the vasculature. The second important thing that I show is that not all the perivascular adipose tissue, that's the one that surrounds the vessels and it's located almost everywhere where there is a vessel of large diameter and medium diameter. They are not created equal. There are different phenotypes. The PVAD, the acronym for perivascular adipose tissue, in the thoracic area is usually brown in rodent models of cardiovascular disease and obesity. In humans, there is some brown adipocytes in patches surrounding the thoracic aorta in the perivascular adipose tissue, and the majority of abdominal perivascular adipose tissue is white. The third thing that I mentioned was that the perivascular adipose tissue has the capacity to sense mechanical stimuli, and that might influence the way new adipocytes are developed. When there is a lot of mechanical stimulation, there might be a reduction in the capacity of those pre-adipocytes, those adipocyte progenitors, to become adipocytes. That's basically a brief summary, a 3-minute summary of my 17-minute talk.
So I thought the session was well attended, and my hope is that people really stayed to listen to advances in adipose biology that they really wouldn't receive everywhere. So I've had the privilege of attending this meeting now for 30 years, and this was a different kind of hour. Intended to be educational. I think the 3 of you did a beautiful job in connecting with one another, from the adipocyte, right, and some of the types of adipocytes, tools you use to study adipocytes, to different fat depots and how they change with aging and blood pressure, to a very special fat that is the perivascular adipose tissue around the vasculature. So I was hugely encouraged, right, by how this hour went. What did you think, Andres? And what were some of the thoughts you had?
Oh, that was an excellent experience if you're in the cardiovascular theme or area, because you often don't hear of these models to trace the adipocyte fate in talks in the Council of Hypertension, for example, because you usually think about the adipocyte as a diabetes, obesity type of topic, but these talks demonstrate that really matters, and matters for hypertension the way the adipose tissue develops, and even the localization of the adipose tissue depots. So I think it was excellent.
So today we held a recent advances in hypertension session entitled Adipose Biology in the Cardiovascular System, Advances, Tools, and Context in Hypertension. I was lucky enough to be in the room when this happened and can say that that session delivered on what the title promised, and that is understanding adipose biology, understanding tools to be able to understand biology, and then how adipose biology impacts the cardiovascular system. Dr. Contreras, Dr. Gliniak, and Dr. Wong were all an important piece of this, also co-chaired by Dr. Clitoria Williams.